Hi, this is Mary from Open Helix with this week's Tip of the Week. If you're seeing this tip someplace else later, be sure to swing back by our blog at blog.openhelix.com, where I'll provide additional details, provide links to the resources that I'm talking about, and the examples in the paper and so on. So check us out at blog.openhelix.com. For today's tip, I'm going to highlight a new data type that's been applied to the UCSC Genome browser. Now, you're probably familiar with the UCSC Genome browser. It's a way to organize and display lots of genomic information that provides context for any region that you might be interested in exploring. But they're always adding new data types. And a new annotation track that's been added is the publications track, and that's what I'm going to show you today. So I'm going to quickly go to a genomic region. I'm just going to click the link for Genome browser here. I'll go to the Human Gateway to start my search, and I'm just going to click a genomic region to quickly explore. But you'll see that this is available in some other species as well, but we'll just click at the human example for now. I'm just going to take you to that genomic region to illustrate the features that I wanted to show you today. So when you arrive at the genomic region here, you'll see here we are in the UCSC genes area that I expected to be in, but you can explore your regions of interest. And there's all the contextual information that you expect to find in the UCSC Genome browser. But in addition, I already have turned on this new track that I wanted to highlight, this track that offers you sequences in articles from now PubMed and Elsevier, and maybe more over time, or SNPs and publications. So a team from UCSC, and also our software was developed in the Bergman lab prior to this as well, they have mined the text that's available from different sources, in this case PubMed and Elsevier, and millions of articles have been searched for sequences that can be mapped to the genome. And they've provided you with tracks here that will let you click on these features, so you could explore any of these articles that you might be interested in. You can learn more about them just by hovering here, as you can see, but you can click on them as well, and you can learn more. Now, this track won't be on when you first arrive and look around at your genomic regions. What you'd have to do is go down to the Mapping and Sequencing Tracks area, and you'll find in this group, Publications here. I'll click on the Publications hyperlink to give you just a close look of what's going on here. So you can explore the publications that have been mapped to any genomic region if you explore this track. So you can turn on the track. You can turn on different ways to look at the features here. We've already looked at the sequences and articles that I mentioned, and the SNPs and publication, but you can see that there might be other types of data you want to turn on as well. So you can learn more about this track. There's description information here on some features about how the display works, and you can learn more about how this was done. There's a methods section here. And this team, and led by Max Hausler here, and he gave me the opportunity to test earlier on, but also the original software came from the Bergman lab here, which is also important. And you can also get information about how this was done from the references section too. So there's a lot of help for understanding what's going on in this track from that track details page. I've already turned on some of these. As I said, you'll have to turn them on yourself. But one of the other things that you can do now is any of the features that you find here, so any of these papers, you can click on these papers and you can learn more about them. So I'm just going to click an example here. And this will tell me more about this paper and what was found in this region. So this might be helpful for you to understand that. But further, you can click on this and you can go to the paper itself. Now, if you have Science Direct, in this case, this is a Science Direct opportunity from Elsevier. If you go to Science Direct, you can also add an app over there. So Elsevier has made the text of these papers available for the team, and they could mine this text, and they developed an app which lets you look at those sequences directly, and you can click from here to go back to the UCSC Genome browser as well. So there's two ways to do this. You could start from the literature if you're looking around at the literature here in the SciVerse, or you could go from any genomic region that you're interested in and explore the literature as well. And what I think is great about this tool is there may be researchers here that were looking for things that weren't what you expect. So some of the features that you're looking at, of course, if you're in a genomic region you're interested in, you'll recognize some of these papers and some of these names. But there may be other researchers who came across this region from other perspectives, and that might help you to understand these genomic regions as well. So I really think this is a cool track that provides a lot of new information that really hasn't been accessible before, the literature information and text mining that. So check out the new publications track on the UCSC Genome browser, and also if you have Science Direct, you can add that app over back at the Elsevier interface, and you can use that to explore the UCSC Genome browser from the literature as well. It's a really neat way to look around. So check these out. Thanks very much for your time.